after that, we move into the post-approval research and monitoring, which is called also the phase 4. So this is the whole drug R&D research processing. From drug discovery through FDA approval, it takes 10 years on average, and a cost average $2.6 billion US. So however, less than 12% of drugs that makes into phase 1 clinical trials is approved by the FDA. So today, like I said, the focus will be on drug discovery process, mainly in neurodegenerative diseases. The drug discovery phase of the R&D process is very critical, as every one of you will acknowledge that. So this involves several phases, mainly 3 steps. The first step of that would be the disease mechanism of action. So to develop a drug, initially you need to know what is the mechanism of action that you're going to target. So once you decide what mechanism of action you're going to target, you identify the MOA to target. So the second process involves drug designing and synthesis. A drug can be as simple as an inhibitor of a target, or an antisense oligonucleotide, or a biologic, such as a humanized antibody. The final step of the drug discovery process, before it moves into the preclinical phase, is screening for therapeutic efficacy. So it can be done through in vitro or in vivo assays, but that should be suitable for the model and also the drug that you're going to test. Being an M.Pharm student in phytopharmacy, I did not have much knowledge about neuroscience until I left JSS. But fortunately, at my PhD institute at University of Strathclyde in the United Kingdom, I was exposed and I was trained in the field of neuroscience during my PhD for 3 to 4 years period. So with that expertise, I would say neurological diseases are primarily affect the brain, spinal cord, and the peripheral nerves. So it leads to significant suffering and disabilities, and it leads to elaborative expenses. And also, there are only few effective drug therapies for neurological diseases. So here are the various neurological diseases and their prevalence distribution. You could see the major part of the pie is shared by Alzheimer's disease, followed by traumatic brain injury, both brain injury caused due to accidents and also sport-related injuries, stroke, epilepsy, chronic pain, Parkinson's disease, multiple sclerosis, cerebral palsy, Huntington disease, and so forth. So my expertise so far has been in these 5 therapeutic areas: Alzheimer's, traumatic brain injury, chronic pain, Huntington's disease, and Parkinson's disease. Today, with the interest of time, I would just concentrate on Parkinson's disease drug discovery. So I don't know how many of you know or study Parkinson's disease. I just wanted to give a basic introduction to Parkinson's disease. Parkinson's disease is the most common movement disorder and is the second most prevalent neurodegenerative disorder, next to Alzheimer's disease. Parkinson's disease affects 1 in 100 people over the age of 60. There are over 1 million cases in the United States and more than 6 million cases worldwide. So it's a really major disease that needs a therapy very soon. Parkinson's disease is pathologically characterized by loss of dopaminergic neurons, which are dopamine-secreting neurons in the midbrain or the basal ganglia. So here you can see the cross-sectional view of a normal brain. You can see the brown staining here is that of neuromelanin. So the area that we have highlighted here is the substantia nigra, part of the midbrain. you can see there is a clear, healthy staining of these neurons in this region. But the figure below, you can see this is the brain section of a Parkinson's disease patient. You hardly see any brown staining. So, which means these neurons have been intoxicated and they are dead. So, their capacity to secrete dopamine and then further processes after that, like motor or cognitive or other functions, will be severely affected. Another major pathology in Parkinson's disease is the formation of Lewy bodies. So Lewy bodies are intracytoplasmic inclusions that contains fibrillar alpha-synuclein, so which we will talk about later in the talk. So what are the symptoms of Parkinson's disease? It's bradykinesia, is the limited movement with your arms and legs. There's limited movement. And resting tremor, so at resting position you will have a tremor, which is the shakiness of your leg or the or the hands. And rigidity, you will not be able to move your arms or legs as you want. And postural instabilities, you'll have an imbalance when you walk. Or even sitting, you have an imbalance. So that's postural instability. So it's a major disease that affects the life of adults or even early onset. So 10 to 15% of this Parkinson's disease is familial, meaning it arises from your genes, from your parents, genes from your parents. It's familial. So if you have a mutation for Parkinson's disease, you are bound to get the disease. So that's only 10 to 15% of the cases, whereas 85 to 90% of the cases are sporadic. Sporadic meaning, so there's no known etiology of this disease. So it might be a gene-related, or it might be gene-environment interactions that has not been known yet. So the therapy for Parkinson's disease, can anyone hear me?
Yes, sir.
Yeah, but I got a message that there was a disconnection.
No, sir. Being participants, we can hear you.
Okay, thank you. So should I continue, Rajendran? Okay.
We could able to hear you, Sudhakar.
Thank you, Muthu.
Yeah, can go ahead, please. Sudhakar can go ahead.
Yeah, thank you.
Red one muted, blue one
Can everyone mute? I'm able to hear some background noise. Sorry. Thank you. So here are the therapies for Parkinson's disease. Based on what treating Parkinson's disease, they can be characterized into two types. The disease-modifying therapy, which is to cure the disease, to target the etiology itself. And the symptomatic therapy, which is to alleviate symptoms such as tremor, bradykinesia, rigidity, etc. So there are also therapeutic categories based on what we target. for example, alpha-synuclein, like I said previously, is a major protein, is a pathological protein in Parkinson's disease. So if we target alpha-synuclein, that drugs will be characterized as targeting alpha-synuclein. And immunotherapy, basically targeting the protein with an antibody or something, or GBA, which is another gene involved in Parkinson's disease, glucocerebrosidase, or neurotropic factors like GDNF, BDNF, or botanicals, which are herb-derived drugs, or cell therapy, which is basically stem cell implantations. So based on what type of mechanisms we approach, there are different categories. So this is the latest pipeline for Parkinson's disease. As you can see, the disease-modifying therapies here, there are many in phase 1, phase 2, and phase 3. As it progresses to phase 3, there is a steep decline in the number of disease-modifying therapies. So this all, enforces the need for a very good drug discovery process. And on the bottom half of the figure here are the symptomatic therapies. There are a large number of drug candidates for this symptomatic therapy, phase 1, phase 2, and phase 3. Even in phase 3, you can see numerous amounts of drugs being trialed. So the boxes here, ambroxol, and then Bond A, which is a botulinum toxin A. So these are a couple of molecules that I was involved in performing a drug discovery process. So here's the list of drug candidates that we had tested for Parkinson's disease therapy during my postdoctoral days at UCLA and University of California, Irvine. So this is a long list of candidates that we tested. They come as different types, like I said, disease-modifying or symptomatic therapy. They target different parts of the disease, I would say, different mechanisms, like targeting alpha-synuclein, GBA, or botanicals, or targeting even the tau, or even APP. So we have also targeted inflammation. So these are the different drugs that we tested with an animal model. Today, I would like to concentrate on the top 2 candidates here, which is CLR01. I would call it Clear 1, and nicotine, which is an ingredient of tobacco. So these are the 2 studies that I will talk about in detail. So I will jump into study 1, where I will talk about Clear 1. So this study was published in Neurotherapeutics in 2017. So we, along with a lab at UCLA, Gal Betan's lab, published this study after 5 years of research on this particular molecule. I'll just get into the details. If you remember, I talked about the drug discovery phase. Like I said, there are 3 steps in the drug discovery phase. Phase 1 is identifying the mechanism of action, if you remember. So here, the mechanism of action we targeted was alpha-synuclein. So with regards to Parkinson's disease, there are multiple genes that have been implicated in Parkinson's disease. As you can see, alpha-synuclein is one of the candidates. And there are other candidates, including LRRK2, other mutations in Parkinson's disease, Parkin and PINK1. There are various genes involved in the Parkinson's disease etiology. But one of the genes that come in both the familial and sporadic Parkinson's disease is alpha-synuclein. So in this cartoon here, you can see alpha-synuclein is a presynaptic protein, which normal function is to facilitate the synaptic processes. what happens in the disease condition is alpha-synuclein aggregates into fibrils. So these fibrils are toxic that will lead to the death of dopaminergic neurons. So these fibrils will enter Lewy bodies. So our intention here in this particular MOA was to target this area where we would inhibit the aggregation of alpha-synuclein. So the second phase, if you remember, is design and synthesis of the drugs. Like I said in the previous slide, we wanted to target the aggregation of alpha-synuclein into toxic alpha-synuclein fibrils. So we collaborated with Dr. Gal Bitan, a professor at UCLA. They designed a molecular tweezer, they call it, which the structure of molecular tweezer is represented here. It was known as Clear 1. This molecular tweezer selectively remodels abnormal protein. Abnormal protein here is alpha-synuclein fibrils. It remodels abnormal protein self-assembly through a reversible binding to the lysine residues. So this is the molecule that we synthesized. I was not involved in any of this process. My role in this project was to screen these drugs in proper animal and in vitro models. So this collaborative work was done by Dr. Gal Bitan at UCLA. And the third step here is to screen for therapeutic efficacy. That's what we did here. So to screen therapeutic efficacy, first we have to identify a suitable animal model or in vitro model, anything that you want to validate. And then the animal model, after identifying a suitable animal model, you validate the animal model. And then after validation, you develop and validate the assays in the model itself. So I will go through these processes step by step in my presentation. So an ideal assay, which a validation assay, which should have target engagement, efficacy, repeatability, and reproducibility, of course. So in the process of identifying an animal model, like I said, we targeted alpha-synuclein aggregation. So we wanted an animal model that overexpresses alpha-synuclein. The reason being alpha-synuclein is involved in familial PD, Parkinson's disease. That's involved in part 1 mutations and part 4 mutations. Part 1 mutation is a single point mutation, and part 4 mutation or triplication of the gene itself. So basically, in part 4 mutation, the patients would have 3-fold alpha-synuclein protein, which might lead to increased aggregation and toxicity. The second reason for choosing alpha-synuclein is Braak's hypothesis, which is a benchmark in any neurodegenerative diseases. So Braak's hypothesis was that the progression of Parkinson's disease begins in the non-dopaminergic structures. That's the brainstem or in the olfactory bulb, which is just above your nose. The brainstem synucleinopathy is postulated to progress rostrally to affect the substantia nigra. Substantia nigra is somewhere in the midbrain, and that will lead to Parkinsonism. And the third reason for choosing alpha-synuclein model is that alpha-synuclein has been associated with high risk for sporadic Parkinson's disease. So based on this knowledge, we selected alpha-synuclein mouse model. So once again, this model was in collaboration with the University of California, San Diego, from the lab of Maslia. So this mouse model, as you can see here, overexpresses human alpha-synuclein, full-length human wild-type alpha-synuclein under the Thyl-aSyn promoter. Basically, these mouse models will have overexpressed alpha-synuclein throughout the brain. And here is the confocal images showing overexpression of alpha-synuclein, human alpha-synuclein in this mouse. You can see here in red, this is human alpha-synuclein staining, synaptophysin staining for the presynaptic terminal marker, and here is the merge. And here we have stained the substantia nigra region. If you remember, in one of the introduction pictures, I was showing the region where there is loss of dopaminergic neurons. So this is the region. This is how the region looks like in a mouse model. So this is the staining of that region in the mouse model. In red, you can see human alpha-synuclein, and green is dopaminergic neurons. So you can clearly see there is co-localization of human alpha-synuclein in dopaminergic neurons. So how to validate? Yes, we have identified this model, but how to validate this model? To validate this animal model in relevance to human form, we considered 3 steps. The first step, does the model have causative factors of human disease, which is alpha-synuclein triplication? We validated this by construct validity. And secondly, are there symptoms and pathology that's seen in human disease, also seen in this mouse model? We validated this by face validity. And then therapy. Okay, what happens when you treat with the same drug that you treat humans? Does the mouse model show any improvement in the disease? So we did this by performing the predictive validity. I will go into this detail. First, construct validity. So for the construct validity, we performed immunostaining for alpha-synuclein to confirm the expression of alpha-synuclein in the brain regions. As you can see, there is 3-fold increase in alpha-synuclein expression, which is similar to the human levels. We performed Western blotting, and we dissected out different brain regions from the mouse model, from substantia nigra to cortex, hippocampus, cerebellum, and olfactory bulbs. In all of those regions, we saw somewhere around 2 to 4-fold increase, which replicated the human disease. Next, we moved on to confirm the face validity. For the face validity, we initially performed pathological assays, basically immunohistology. If you remember, I talked about Lewy bodies. Lewy bodies are cytoplasmic inclusions of alpha-synuclein. So on the left here, where my cursor is the brain section of a Parkinson's disease patient. You can see there are black spots or dark brown spots, which are Lewy bodies. And these are Lewy neurites, which are the nerve endings of the dopaminergic neurons. Similarly, on the right-hand side of this picture, you can see this is the mouse model staining of Lewy bodies. Yes, you can find similar pathology in this mouse model, and also Lewy neurites here. That was confirming that the mouse model has a similar pathology to the human disease. We looked for the pathogenic marker, which is seen in human disease. That's the phosphorylated S129 alpha-synuclein. We saw that in the mouse model. And also we looked for inflammation in the mouse brain, which we also see in the human brain. We stained for microglial activation, which is by IBA1 staining. As you can see here, there are inflamed or activated microglia. In the second step of pathological assay, as I said, there is loss of dopaminergic neurons, and there is decrease in the dopamine levels. But this all happens in the later stage of the disease. What happens if a person gets the disease much early? So between 25 to 50 years of age, there is a high dopamine turnover in Parkinson's disease patients. It's only after 60 years of age there is a decrease in dopamine levels, leading to motor dysfunction. So is this the same in the mouse model? And what we found was astonishingly similar. Yes, the mouse model showed increased dopamine levels at 3 months of age, which is an young age for a mouse model. which is equivalent to 30 to 40-year-old, I would say, or 20 to 30-year-old, I would say. And the old mice, which is 14 months, which is closer to 60 to 70 years of human age, you can see there is a decrease in the dopamine levels. So yes, the neurotransmitter levels were also similar to the human disease. So the next major symptoms in Parkinson's disease is the behavioral symptoms. As you can see, the motor behavior is the major deficit in Parkinson's disease. In addition to motor behavior, there are other deficits as well, which includes the cognitive behavior and also social interaction. A person with Parkinson's disease may not interact with other people as normal people. That's social interaction. And the person with Parkinson's disease will also have anxiety. So how to test this in the mouse model? We performed a battery of behavioral assays, which I will run through very quickly here. We performed behavior assays. Here is an example of a motor behavior assay. So this is called the challenging beam test. So on day 1 and 2, for 2 continuous days, the mouse would be allowed to walk on a beam which narrows and tapers to a tiny rod at the end. So this will happen for 2 days. So after 2 days of training, on the 3rd day, we would place a grid on top of this beam, and the mouse would be allowed to walk on this beam. So this is a picture of how the grid looks like. So as you can see in this red circle here, there is a slip of the paw through the grid. So this is due to the loss of gripping strength in the mouse. So it slips through the grid. So we calculate this as an error, and the errors are calculated based on the number of steps they take on the grid. You can clearly see there is an increased error in the Parkinson's disease mice. So this is at different ages. At 2 months, 4 months, 6 months, and 8 months of age, you can see there is also a progressive increase in the motor deficit. The second test for testing the motor behavior we performed was the pole test, which is the test where we let the mouse sit on top of a pole, and then we calculate the time required to turn and traverse down the pole and reach the bottom of the pole. So that's called the time to traverse. You can see, once again, the Parkinson's mice taking longer time due to their motor deficits to get down to the bottom. There is a progressive increase here as well. So moving on to the cognitive behavior, we performed the 3 familiar assays in cognitive behavior. The first assay we performed was called as novel place recognition. It's basically testing the memory of the mice on the place preference. With trial 1, in trial 1, what we do is place 2 identical objects in a Y maze and allow the mouse to explore for 5 minutes. And after the mouse explores for 5 minutes in trial 1, in trial 2, we switch one of the objects to the other arm. What we calculate here is how the animal is able to discriminate the place. Is it staying more in arm A or arm B? And we calculate that as a discriminative index. As you can see here, in Parkinson's disease, there is a deficit in this discrimination index. This is Y maze, another cognitive test. This is an open Y maze. You leave the mice in here, allow the mice to explore for 10 minutes. And then basically you calculate the alternations being alternative arms. So it should go from A, B, C, or C, B, A. It should alternate and then have a pattern. So if it has a pattern, then we give it a score. And you can see the wild type mice performing really well, 60%, whereas the Parkinson's mice doing not that well. And the third cognitive test that we performed was novel object recognition. Here, what we would do is in trial 1, 2 identical objects for 5 minutes. And then trial 2, you switch one of the objects with a novel object. And we calculate the discriminative index. So here, the wild-type mice explore the novel object more, whereas the Parkinson's mice don't. So that's a cognitive deficit. Moving on to social behavior. It's a 3-chamber test. Basically, there are 3 chambers in here. It's an open chamber. The mouse model in trial 1 and 2, the mouse model can freely move around. In trial 2, there are 2 cups placed on the left and the right hand, on the left and the right chambers. In trial 3, what we do is place a novel mouse, which this mouse, the testing mouse, has not encountered. And then we would see how this testing mouse would interact with the novel mouse. So the wild-type mouse pretty much spends a lot of time sniffing the novel mouse, whereas the Parkinson's mice do not interact socially. This is something that I previously talked about, the dopamine levels. We saw in a similar pattern to the human disease. So an increase in dopamine levels will basically increase your anxiety, both in humans and also in mice. So we wanted to test this phenomenon. So we used the open field behavior testing. You can see in young mice, as I said, there is increased dopamine levels. So there is anxiety and there is increased locomotion. You can see there's a lot of movement for this particular mice. And in older age, the same mice shows decreased movement in the open field. So that's where we confirmed, yes, this Parkinson's disease mouse model has similar behavior phenotype to the human disease. So one other a major thing in Parkinson's disease is mitochondrial dysfunction. So there is evidence that supports mitochondrial dysfunction and increased oxidative burden in Parkinson's disease pathogenesis. We measured mitochondrial function using a seahorse analyzer, which is an instrument that measures oxygen consumption rate. So what we did was we isolated the brain regions from the mice and quickly isolated mitochondria from this tissue and put into this seahorse analyzer and performed 2 assays, the coupling assay and the electron flow assay. And what we saw here was a severe deficit in a mitochondrial complex activity. We also confirmed this by performing an ELISA for complex 1, that's mitochondrial complex 1. You can see in black, you can see the Parkinson's disease mice showing a significant decrease in complex 1 activity. So overall, what I've shown here is the mouse model shows construct validity compared to the human disease, and also the phase validity compared to the human disease. So the third part of the validation is, so what if we treat the mouse with an available therapy like L-DOPA or dopamine agonist? That is what we did in predictive validity. Basically, we administered L-DOPA, which is a therapy, a symptomatic therapy for Parkinson's disease, in older alpha-synuclein mice, and we performed an adhesive removal test. Basically, the test is you place a sticker on the nose or the snort of a mouse, and we calculate the time required to remove the sticker. So this data here shows that wild-type Parkinson's mice treated with saline shows normal removal time, whereas the Parkinson's disease mouse show a deficit. They take longer time to remove the snort. So overall, we validated this mouse model for comparisons to human disease, and yes, this mouse model is valid and can be used for screening drugs. So now I will get into the actual preclinical trial, which is the more interesting part, I would say. So if you remember, the first step of the drug discovery was identifying the mechanism of action. We identify alpha-synuclein aggregation. To inhibit alpha-synuclein aggregation, we designed and synthesized a molecule with the help of a collaborator. And here is our model, which we validated. Now we are going to test this particular drug in this model. Okay, so how do we administer the drug? We administer the drug centrally, that is into the brain directly. This is for a proof of concept. What we did was we packed the drug into an osmotic pump, which is connected to a metal catheter. And we performed a stereotactic surgery on the mouse. We drilled a hole in the skull that targets the lateral ventricles, which is the pathway by which there is the flow of cerebrospinal fluid into the brain. So we targeted this lateral ventricle, and we inserted the cannula into this particular area. And after insertion, we placed the osmotic pump into the subcutaneous pocket. We inserted the catheter into the brain coordinates, and then the drug was delivered for 28 days. That's 4 weeks of drug delivery. And after that, the drug package was disconnected, and we performed all the behavior tests and histopathology that I mentioned earlier. So based on the data that we collected from this particular study, you can see here before treatment, there was no difference in this mouse model. So before treatment, there was nothing, so everything stayed the same. But after treatment, after 4 weeks of treatment, you can see there is an improvement in the motor deficit with the lower dose. So what happens to alpha-synuclein levels in the brain? This is a depiction of the brain regions that we were interested in the Parkinson's disease mouse. So this is the striatum, where you have the dopaminergic nerve terminals. So this is the substantia nigra, where you have the cell bodies of the dopaminergic neurons. So what we did was we isolated tissue from this particular brain region and performed Western blotting for alpha-synuclein levels, and we quantified the alpha-synuclein levels. What you see here in the striatum especially, there is a decrease in alpha-synuclein levels. So this particular experiment shows that yes, our drug has worked and it has reached the target. There is a decrease in alpha-synuclein levels. What happens to the aggregates and oligomeric forms of alpha-synuclein? We performed immunohistology. The drug that we administered, that's CLEAR-01, at this particular dose did not modify the aggregates or oligomers. We also confirmed that with a native gel Western blotting in the striatal and substantia nigra regions. So we didn't see any change in the aggregates or oligomers, but we saw a decrease in total alpha-synuclein levels. In addition to central administration, we also administered CLEAR-1 peripherally meaning subcutaneously. If you remember, so this was the osmotic mini pump that had a connection to the brain in the previous drug delivery method, whereas here we placed this osmotic pump into the subcutaneous area and then delivered the drug subcutaneously. So here is the data from peripheral CLEAR-1 administration, challenging beam test, improval of the motor deficit, in the challenging beam test, and also in the vertical pole test, you could see there is an improvement in the motor deficit. In summary, this particular drug trial, where we administered clear one centrally and peripherally, there is an improvement in motor behavior. There is decrease in striatal alpha-synuclein levels, and also motor deficits were improved in peripheral administration of this drug. So this data supports further development of this clear one drug as a therapeutic agent for Parkinson's disease and other synucleinopathies. With this, I will move into the next study. If you remember, I wanted to talk about two studies. The first one being CLEAR-01, and the second one is the nicotine, which is an active ingredient in tobacco. So this is a very interesting study. I would not advise everyone to smoke. This is I'm showing neuroprotective effects of nicotine, but this does not promote smoking. So nicotine is part of tobacco, but there are several other toxic ingredients in smoking. So this paper, we published this paper in 2018, I guess. Yeah, 2018. This was in collaboration with Caltech and Michael J. Fox Foundation for Parkinson's disease in New York. So this study was funded by Michael J. Fox Foundation. So this is a very interesting target, what do you say, target discovery. So why would you select nicotine as a target? So we digged into the epidemiological studies and we found out there is a strong link between smoking and decreased incidence of Parkinson's disease. That was very surprising. There are several papers that have reported this. So what happens when you smoke? The risk of Parkinson's disease in current or a former smoker is about half that of a never smoker. Okay, that was an astonishing data. So we looked into this data very closely, and in consultation with several experts in this field, we decided to test the neuroprotective effects of nicotine. So we dug into the literature and we found that nicotine on its own can protect against toxicity induced by several neurotoxins in dopaminergic neurons. So that was very interesting. And we found out that nicotine actually targets alpha-4, beta-2, and alpha-7 nicotinic acetylcholine receptors in the brain. And guess what? So these receptors are present in the substantia nigra, so which is the region of interest, the region highlighted here in green, so which is the region of interest in Parkinson's disease. You can see alpha-4 beta-2, yes, it's present in the substantia nigra. Alpha-7, yes, it's present. So nicotine actively targets these receptors. So we gave a proposal to Michael J. Fox Foundation, and then they accepted the proposal, and we started the study. Yeah, so we performed two different studies. One, a chronic exposure, meaning exposed to nicotine for a longer time. So in mouse age 6 months is a chronic exposure. We exposed nicotine for 6 months continuously in the Parkinson's mice that I described earlier. And the second study was 1 month exposure of nicotine. And we performed the behavior and then immunohistology tests, like I mentioned before. So how we delivered the drug was once again in the osmotic mini pumps. We loaded the drug, primed the drug for 48 hours, anesthetized the mouse, prepared the mice for surgery, created subcutaneous pocket, and insert this subcutaneous osmotic mini pump subcutaneously, and then suture or clip the incision and apply local anesthetic. So this the drug will be delivered for 4 weeks. This is what we did for the 1-month cohort, and for the 6-month cohort, we basically performed this 6 times on the same mice, meaning we performed the surgery 5 more times after the initial surgery. This was a pretty intensive study that we performed. So here are the results, the primary results, the major findings from this result, from this study was 7 months, at 7 months of age. What I show here is the wild type mice treated with vehicle. They show alternations, normal alternations in the Y-maze test that I described earlier, whereas the vehicle administered mice showed decreased alternations, and then treatment with low and high dose of nicotine actually improved the alternations, showing there was an improvement in the cognitive deficit, which almost normalized to the wild type levels. We performed the other test, that's the novel object recognition test. Once again, administration of nicotine showed improvement in the discrimination index, showing there is clear improvement in cognitive deficits in Parkinson's mice. Remember, I talked about social behavior. Parkinson's patients and also Parkinson's mice show deficits in social behavior. In this particular test, when vehicle-treated mice showed a deficit in social behavior, and when you treat with nicotine, there is an improvement in social behavior. And what happened to motor deficits? We did not see any improvement in motor deficits. However, like I showed previously, there was a huge improvement in cognitive and social behavior. So overall, this is a summary. Peripheral nicotine treatment reverses cognitive deficits and social impairment. And nicotine treatment did not alter motor deficits. And this kind of supports that nicotinic acetylcholine receptors, agonists for nicotinic acetylcholine receptors may improve cognitive and social deficits in early Parkinson's disease. So this is an example of a symptomatic treatment. You can basically treat patients with nicotinic agonists for cognitive deficits. So with that, I want to give you a summary of preclinical trials that I was involved in the last 13 years. So I would like to go through this list slowly so that I can capture the whole idea behind this. So here are the drug candidates. The data I showed for nicotine drug discovery testing, and clear 1 drug discovery testing. So the mechanisms here were agonists for the receptor, prevent alpha-synuclein aggregation. We performed these tests in Parkinson's disease model. I was involved in another study, which is in phase 2 clinical trials, where we tested ambroxol, a drug that modulates glucocerebrosidase, once again in Parkinson's disease. And we performed antisense oligonucleotide testing, which targets the mRNA of alpha-synuclein, or in Huntington's disease, it would target the Huntington mRNA. We performed that study in this model as well. Isofagamin, which once again modulates glucocerebrosidase. We performed that test in Parkinson's model. And posifen, which is an inhibitor of amyloid precursor protein, which was a main therapy in Alzheimer's disease. We also tried that therapy in traumatic brain injury. And we used an antipsychotic drug from the company Sumura, Japan. We tested that in Parkinson's disease. We used another drug called devunatide, which inhibits tau phosphorylation. We tested this in Parkinson's and also Alzheimer's disease mouse models. In addition, we tested a drug, we called it Z, also in combination with X. This is a repurposed FDA-approved drug for anti-inflammatory and neuroprotective effects. We tested this combination in several mouse models and in vitro models, such as Parkinson's, systemic inflammation, such as chronic disease, colitis, ulcerative colitis, or Crohn's disease, gut inflammation, and also in traumatic brain injury and Alzheimer's disease. And finally, I also tested, I was also involved in testing biologics, I would call it A, B, and C, for proprietary reasons, so which these biologics inhibit neurotransmitter release, pain neurotransmitter release, like CGRP or neuropeptides. We tested this in chronic pain models. So with this, I would like to end my talk. I would like to acknowledge my mentors and colleagues at UCLA, where I did my postdoctoral work. It's a beautiful place, a wonderful institute, and I've got a great mentor in Marie-Francois Cheslet, who is one of the major names in Parkinson's disease. And we collaborated with Caltech, Dr. Henry Lester's lab. Then also thanks to Dr. Fedorov, who I was part of his lab at the University of California, Irvine, and the people who worked there, it was tremendous. And the lab where I did my PhD, which is in Scotland, Glasgow, was a great place once again to train, and awesome people there as well. Dr. Elizabeth Ellis was my mentor for my PhD, where I learned a lot of neuroscience there, and the scholarships that helped me to do my PhD. And here are the funding agencies that supported all the clinical, all the preclinical tests that I mentioned earlier. With that, last but not the least, I cannot end without mentioning JSS College of Pharmacy, UTI, and the role it has played in shaping me as a person and my research career. Proud to be a JSS College of Pharmacy alumni. If anyone wants to reach out to me, I'm happy to hear from you. Here's my email and my LinkedIn link here. Thank you for your attention. I can take any questions now.
Good morning, sir. May I ask a question?
Of course.
Yeah, my name is Deepak, sir. You spoke about that stereotactic surgery, and through that route, you have given the drug to the brain, and you wanted to see the effect. Okay, when you're reaching the ventricle that way, now the stereotactic surgery itself is a, you can say, concussion to the brain or an injury, and which can increase the level of alpha-synuclein itself. In that case, how do you quantify the actual induction of alpha-synuclein as a result of that stereotactic surgery? And if that is not quantified, then how do you exactly claim the significance of a drug to treat Parkinsonism by suppressing alpha-synuclein level or anything? That is the question for me, sir.
Very good question. Yeah, normally what you're suggesting is the stereotactic surgery itself is a traumatic brain injury, and there are evidences that there is increase of these toxic proteins in those circumstances. Yes, it's a valid point, but we have appropriate controls. For example, we have mice treated with vehicle, administered vehicle via the same route. route of administration, and we have wild type groups, which we have been monitoring all along, right? So we don't only do immunohistology or behavior on one particular group. So we performed all the behavior tests, immunohistology, alpha-synuclein quantification in all of those groups. So what we found nothing abnormal. Our surgery did not increase any alpha-synuclein in this model, and we did not see any such instances, even in other preclinical trials that we were doing. So basically, we are reaching the ventricles. Yes, we are damaging the skull, but that would not tremendously increase the alpha-synuclein levels. So like I said, we had control groups, and the control groups didn't show any such increase in alpha-synuclein.
That is no increase at all you're saying, sir?
No.
Okay, sir. And sir, I have one more question. Like
Sure.
often we read about overexpression of the cholinergic component of the brain, and along with a deficit of the dopaminergic component. To what extent actually the patient suffering from Parkinsonism show an overexpressed cholinergic component? Because there are drugs like benzexol and piperidine, which is theoretically present in the book. But I'd like to know what is the exact percentage of patient who show with an overexpressed cholinergic component.
Yeah. So that's another good question. You're asking about the imbalance. So when we studied Parkinson's disease, we always know that there is a decrease of dopamine and increase of acetylcholine. That's what we study. But from what I understand, and the literature suggests, yes, anticholinergic therapy can improve symptoms in Parkinson's disease. Yes, that can definitely decrease the rigidity and the continuous motor stimulation that you have in Parkinson's disease. Yes, but nothing can match the substitution of dopamine. So you should basically increase the dopamine levels. Yes, so increasing dopamine levels to a certain point works. Like there is a phase in Parkinson's disease patients when you treat them with L-Dopa. Yes, they respond very good for a certain number of years, right? So after a few years, when they reach the level, they may not respond to L-Dopa therapy again. So that basically the L-Dopa therapy will show the opposite effect, meaning they will show more tremors, more rigidity after L-Dopa. So there is what you would say that there is a response, and there is an opposite response to that particular drug. So your question was, so to what percentage respond to cholinergic therapy? Same as
sorry to interrupt. My question was like, what percentage of patient population actually show an overexpression of this cholinergic component? Because those drugs are there, but what is the practical utility? That I wanted to know, because they have not mentioned what percentage of people actually show an overly expressed cholinergic component. So that is the thing. Suppose 100 patients are there for Parkinsonism. How many of them show an overexpression of the cholinergic component?
Yeah, basically, I would say cholinergic component. I think cholinergic component you're saying is the neurotransmitter acetylcholine, right? So acetylcholine, yes, I would say there is increase. I think almost all patients are showing decrease in dopaminergic in dopamine or dopaminergic neuron death would show increased acetylcholine levels.
Okay, sir. Thank you.
No problem.
The patient stop responding to the L-Dopa therapy. Bromocriptine is the choice. It seems that again sensitizes the patient towards the L-Dopa therapy. So it's kind of rekindling. So is it truly practiced?
Yes, I think it depends on patient to patient how they respond to therapy. but initially for any patient, they start with L-DOPA therapy, yes, and then if it doesn't work, they try for other neurotransmitters or dopamine receptor agonists are tried. Yeah, so that's the problem with treating the symptoms, right? So basically, there has to be more disease-modifying therapies rather than symptomatic therapies.
Okay, so thank you so much. It was a very nice presentation. We learned a lot. Thank you so much, sir.
You're welcome.
Yeah, Tibras.
So if you're talking, I can hardly hear you.
Yes, sir. Now in the middle, the connection got interrupted, sir. So it's now I can listen. Hello.
Yeah. Did you ask me something?
No, sir. Thanks. Thank you so much.
Okay.
Thanks for the.
Rajendra, are you still there? Yeah. Hello, Rajendran, can you hear me?
இது
Hello, Rajendran, can you hear me?
Dr. Sudhakar?
Yes.
This is done for Sudhakar.
Hello, sir.
How are you, Sudhakar? Good to see you. Good to see you.
Good to hear from you, sir.
Yeah, thanks for coming on board and engaging our students, research scholars, and also outside the university students. In fact, it was a really good talk. First half an hour I could not be there because I had some other commitments. Okay, last half an hour I was listening to your thing and also the question and answer session, and it was I think very well received by the participants. So on behalf of Swamiji and university officials and also our staff and students, I'd like to thank you for accepting our request and then, coming on board as a speaker to speak out on the topic, preclinical trials. And I had to thank Dr. Rajendran, who is coordinated with you for the last 1 week, and then, bringing you, and then, to deliver this lecture. We are very much proud of you as our proud alumni of our college. And I'm very happy to see you at this stage, at this elevated level with the good knowledge and then, sound research profile and your flow of your language. We are very, very happy, which was lacking earlier in about 15 years back when we were in the department, Just I'm really seeing a very good researcher, and as academician, Dr. Sudhakar. Really, we are very, very happy, Sudhakar. And we feel proud of you. Yeah.
Thank you very much, sir. It's always a pleasure talking to you and JS's students. Anytime, if I can give a talk anytime.
Dr. Sudhakar and Dr. Doiswami, can you hear me?
Hello, sir. Doiswami, sir. Happy to
Yeah, fine. As a teacher, I am very proud of you. It was very nicely presented. And you have explained everything about the Parkinson's disorder. And I think it was a very good decision. And the last point I would like to mention is we have told about the nicotine, which will reverse the pharmacogenic disorders. That is a very pleasant news to me. And,
many.
Nice to see you this level as mentioned, and happy we will have one more day, one more session of yours with some other topics also in the coming days. Thank you very much.
Sudhakar, during our next visit to India, hopefully after 5-6 months and after this pandemic over, okay, you are welcome to our campus and also interact with our staff and students for 1 or 2 days. As a special guest, you be with us and then, interact with our staff and students, and it will be our pleasure to have you, okay, in the campus, okay, during your next visit of your family note to India, okay?
Yes, sir. Sure.
Okay, I think you are.
Thank you so much. Thank you. Thanks for your presentation. And from the Department of Pharmacognosy, we thank every participant who came here and attended the webinar. And
Thank you. I didn't hear anything you said, but still, thank you.